Thank you for the introduction. So I'm going to be talking about how we can map the mechanical properties of the brain and the development of a standard MRE template. So maybe not everyone here is familiar with MRE, and it's a non-invasive MRI-based technique that is sensitive to the viscoelastic or the mechanical properties of the brain. So the procedure generally involves 3 steps. The first involves introducing mechanical waves or shear waves into the brain, usually around 50 or 60 hertz. This displacement information is captured with a motion-sensitive imaging sequence and converted to mechanical properties such as stiffness using a finite element solution to the wave equation. So shear waves travel more rapidly in stiffer materials, resulting in a longer wavelength, and slower in softer materials. That's a little bit exaggerated. It's not actually as bad as that. So you might be wondering why we care about the stiffness of the brain, and the answer is because these properties act as proxies for brain tissue microstructure. And we know that these properties are affected due to aging, in neurological conditions, and this could be tumours or diffuse diseases such as Alzheimer's or MS. And MRE has also been related to cognitive functioning in healthy participants. So there's been work that's looked at the hippocampus and its role in relational memory. And also MRE measurements have been corroborated with biological processes in animal models of disease. So generally there are 2 output parameters, and these are the shear stiffness and the damping ratio. The shear stiffness is a composite measure of the shear modulus and describes the effective stiffness at the actuation frequency. And this is computed based on the wavelength of the waves as they travel through the brain. The damping ratio is a measure of the relative viscous to elastic properties, which describe the dissipative behavior of the tissue determined by the cellular network. And this requires the measurement of the wave attenuation. So the higher the damping ratio, the more the tissue is like a viscous fluid and less like an elastic solid. So MRE is a rapidly growing field. Typically, most studies have recruited roughly 10 to 20 people, sometimes 50, but that's quite rare. And the focus of these studies has mostly been technical developments within engineering, where they've looked at the global brain or a limited number of structures. So in this study, we wanted to provide a complete and comprehensive understanding of viscoelastic properties of all brain regions, using a large number of subjects that are not normally studied within the field. So just to go through the methods that we used, we compiled data from multiple studies, and all this data was collected on a 3T Siemens scanner at 50 hertz. We used a multi-shot, multi-slab spiral imaging sequence, either a 1.6 or 2 millimeter resolution, and data was analysed with non-linear inversion. So this is a subzone optimization approach where you iteratively update properties until displacements agree with an initial model estimate. And these type of models allow for spatially varying properties. We visually inspected all the images, and we co-registered the MRE images to standard space using advanced normalization tools. So here you can see on the left is a standard to 2 millimeter template. The image in the middle is the grayscale version of the stiffness map, which is shown between 1 and 5 kilopascals. And the middle and the right image are the same, just with the color scale. And you can see that there are distinct regions of higher stiffness, lower stiffness, and these are quite symmetrical. So I should mention that was the mean, the average of all the 134 subjects. So when we looked at the global results, so the white matter, the subcortical grey matter, the cortical grey matter, we see that there's differences between the 4, which we'd expect, and that the subcortical grey matter is actually a lot stiffer than white matter or the cortex. And roughly the global brain stiffness is 2.5 kilopascals, which is what was concluded from a meta-analysis. We didn't observe a region times sex interaction. Moving on, we looked at the subcortical grey matter. We found that the pallidum and the putamen were a lot stiffer than the other parts of the deep grey matter. The hippocampus was the softest. More importantly, that they are each distinct from each other. But we did observe an interaction whereby the female brain actually had a lot stiffer hippocampus compared to the male brain. So what this means, we're not sure just yet. Well, no, I won't go into it. Then we looked at the white matter tracks, and again, we saw a significant effect of region. They all display distinct properties. Again, we see a interaction where there's differences between male and female brains. Again, the cingulums involved the hippocampus. We also looked at the cerebral cortex, and we've grouped these by labor regions. And again, a significant effect of region on stiffness. Another significant interaction between these regions. So these were all the stiffness kind of results. I just briefly went through them because there's a lot of information there. But I also talked about the damping ratio, which is the measure of tissue organization. And you can see it here, that the brighter spots are more viscous properties of the tissue. And this is interesting because this is the parameter that's routinely been associated with cognitive functioning. And moving on just to look at the global measures of this is really quite variable. So even though they're quite distinct in terms of the global damping ratio, the white matter, the subcortical, we can see a huge variation, and that's what's particularly interesting, I think, for when we want to look into more cognition-based tasks. So to conclude, the brain regions differ in the mechanical properties or their viscoelasticity. It complements previous MRE results with smaller sample sizes. That we can see evidence of sex differences in certain regions across the cortex, the white matter and the subcortical grey matter. We know that the damping ratio is much more variable, but that's likely to reflect normal individual differences. We're interested in actually characterizing the large-scale brain networks, so we'll be doing that in the future. And we want to make this data and the MRE templates made available for future analyses. So I just want to thank my lab, and I'm happy to take any questions. Thank you.